Hello everyone. Myself PRAJWAL. I'm studying in second year MSc Microbiology at CUTN Tamil Nadu. In the course Marine and Environmental Microbiology, instructed by Dr. Kavya ma'am, I'm taking topic on microbial degradation of environmental pollutants, precisely focusing on petroleum hydrocarbons. So let's get started. Petroleum hydrocarbons are major pollutants. They're toxic to ecosystems and can contaminate soil and groundwater systems. They enter the environment through oil spills, industrial activities, and leakages. They persist in soil and water, causing ecological damages. Microorganisms play a key role in natural degradation of these petroleum hydrocarbons. So, usage of these microorganisms is actually a basis of bioremediation technology. So, what are petroleum hydrocarbons basically? Petroleum hydrocarbons are complex mixtures of organic compounds primarily made up of hydrogen and carbon. They are derived from crude oils, which is an essential for forming fuels like gasoline, diesel. There are several types of petroleum hydrocarbons exist, and there are several parameters also considered for these several types of petroleum hydrocarbons. But here I'm explaining types of petroleum hydrocarbons based on chemical structures. First type include alkanes, also known as paraffins. They are saturated hydrocarbons, exist in straight or branch chains. Examples include methane, propane, waxes. They are relatively stable. Then coming to next type, aromatics. They are ring structured with decolorized electrons like benzene, toluene, xylene, and polycyclic aromatic hydrocarbons. They are often considered as carcinogenic. Then the next type, cycloalkanes, also known as naphthanes. They are saturated rings. Examples include cyclohexane, and they are very less reactive than aromatics, but they are more reactive than parabens. The next type, it's actually a uh, compound based on the composition like nitrogen, sulfur, or oxygen. They are also known as heteroatomic. Examples include thiophene and phenols. The last type called asphaltenes. They are large, complex molecules, often solid or semi-solid states, and they are found in heavy crude oil. So, what are the sources of this petroleum hydrocarbon pollution? The sources are oil spills, accidental releases from tankers, offshore platforms, pipelines, and storage tanks during the extraction, transport or storage process. From vehicle and transportation, leaking oils, fuels, lubricants from cars, trucks, and machinery washed into drains, and they connect to waterways by rain. And another source include industrial and manufacturing, where waste from oil refining, petrochemical plants and facilities using creosote or coking processes may also contribute to petroleum hydrocarbon pollution. And other two sources are urban runoff like a stormwater carrying drips from vehicles, grease, and other fluids from streets and parking lots. So, what are the environmental and health impacts of these petroleum hydrocarbon pollutions? Environmental impacts include soil and water, which the soil quality degrades and restricts the water or nutrient flow, contaminates the groundwater, and harms water soil microbes. So they also inhibit seed germination, reducing photosynthesis and limits the nutrient or water uptake in plants. And the most lethal to aquatic lives like fish, whales, sharks. They're the most targets in aquatic lives. Then wildlife. They causes a poisoning, reproduction failure, and a death in birds and mammals. Coming to human health impacts, we have exposure routes like inhalation or ingestion and skin contact, through which we could acquire some health hazards like for short-term hazards include dizziness, headaches, fatigue, respiratory irritation, nausea, or skin eye irritation. Long-term includes cancer, like a benzene in gasoline is linked to leukemia causing. Then it also causes some vital organ damages like heart, kidney, liver failures. So there are major hydrocarbon degrading microorganisms. I have given some examples for bacteria like Pseudomonas, Alcanivorex, Actinobacter, and Bacillus species. For fungi, it includes Aspergillus, Penicillium, Candida, and Phanerochaetae. For actinomycetes, it includes Rhodococcus, Mycobacterium, and others. So the general process involved in this degradation of petroleum hydrocarbons are as follows. Microbes secrete surfactants to break down these large oil droplets into smaller ones, increasing the surface area. And adaptation. The emulsified hydrocarbons stick to the surface of microbial cells, and these hydrocarbons are transported into the cell through active or passive transport, or also endocytosis. Then from there, the next step includes enzymatic breakdown of these compounds. Inside the cell, enzymes like alkane hydroxylases and oxygenases catalyze these reactions that break down the complex hydrocarbons chains into simpler molecules. The broken down molecules are fed into central metabolic pathways like TCA cycle to generate energy and building blocks for cells growth. The final products include these compounds are at end they are converted into carbon dioxide and water. So there are two types of degradation based on the presence or absence of oxygen. The first type includes aerobic degradation of hydrocarbons, which means this process takes place in the presence of oxygen. Here, the initial oxidation of microoxygenases and deoxygenases takes place. Alkanes are converted into alcohols, and alcohols are converted into aldehydes, and aldehydes to fatty acids. And fatty acids enter the beta oxidation pathway, finally oxidized via TCA cycle. And the next type, anaerobic degradation of hydrocarbons. Here, the whole process takes place in the absence of oxygen. The main difference between these two types are the terminal electron acceptor. In aerobic degradation of hydrocarbons, the terminal electron acceptor is oxygen, whereas in anaerobic degradation of hydrocarbons, it is alternative electron acceptors like nitrate, sulfate, or iron. So, but the disadvantage compared to aerobic degradation of hydrocarbons, it is it is more slower. And the advantage is they are important in sediments and deep soils. The factors affecting microbial degradations: the oxygen availability, nutrient availability, like excess amount of nitrogen, phosphorus, if it is present it may enhance the degradation efficiency of the microbes. Then the pH and salinity, optimum pH and salinity is important, and the chemical nature of hydrocarbons, whether they are breakable or unbreakable, that is also matters. Then the presence of suitable microbial populations also affects the microbial degradation. Then there are several steps for enhancing this microbial degradation, like biostimulation, adding a nutrients, as I told, nitrogen, phosphorus, and vitamins to the contaminated site, so that we could encourage the growth of the native, means indigenous micropopulation, and they can increase the rate of degradation process. Then bioaugmentation, introducing a specific pre-selected or genetically engineered microorganisms that are particularly good at degrading hydrocarbons. Another method includes combining the biostimulation and the bioaugmentation can be more effective as seen in studies that used nutrient addition alongside a microbial consortia. So I have added some, actually one real incident. Exxon Valdez oil spill. This accident occurred in Alaska, USA on March 24th, 1989, when the tanker hit Black Reef in Alaska's Prince William Sound, spilling 10.8 million gallons of crude oil, devastating wildlife, and 1,300 miles of shoreline. The environmental disaster included over 1,300 miles of shoreline were oiled, then the massive wildlife deaths. For example, 250,000 seabirds were died in that accident, and the economic devastations, like fisheries, are closed, impacting a local economics and tourism, and also the cleanup and cost. Exxon spent over $3.8 billion on cleanup and paid billions more in fines and settlements. So they also used bioremediation. For bioremediation, they used nutrient fertilizers to enhance this degrading process. It's one of the landmark case for large-scale bioremediation. So what are the advantages of microbial degradation? They are eco-friendly and sustainable, cost-effective compared to physical methods, complete mineralization possible, and minimal secondary pollution. Very less secondary pollution it is produced, and it is also suitable for in situ treatment. What are the limitations of microbial degradation? It includes slow process, ineffective for highly recalcitrant hydrocarbons. The environmental constraints affect its efficiency. It requires continuous monitoring and optimization. The future perspectives include genetic engineering of these hydrocarbon degrading microbes, so that the engineered metabolic pathways can result in a faster degradation of these petroleum hydrocarbons. Then use of microbial consortia instead of single strains, application of omics technologies, and integrated bioremediation approaches like nanobioremediation technology combined with other bioremediation technologies. So, in conclusion, petroleum hydrocarbons are major environmental pollutants. Here, microorganisms play a crucial role in degradation. Real oil spill events prove that effectiveness of microbial action. Bioremediation is a promising and sustainable solution. Thank you.